Good afternoon, everyone. I'm Steve Katz, Director of the National Institute of Arthritis and Musculoskeletal and Skin Diseases, and I'm delighted to host Dr. Roy Bar-Ziv for the WALS lecture today. Roy did his undergraduate studies in physics and mathematics at the Hebrew University in Jerusalem and his graduate studies in condensed matter physics at the Weizmann Institute of Science in Israel. We don't have too many physicists come and give the WALS lecture, so Roy is going to bring physics to biology and biology to physics. Let me tell you a little bit more about Roy. He did his postdoctoral fellowship at the Rockefeller University in Albert Liebhaber's lab, and Roy made a transition to biological physics at that point. He used sensitive fluorescence measurements to study in vitro how proteins involved in recombination polymerize on DNA. And at the same time, he became interested in the subject that he's going to be talking about today, in studying gene regulation, and worked on the first attempt to assemble cell-free gene expression circuits. In 2003, Roy returned to the Department of Materials and Interfaces at the Weizmann Institute as a PI, where his main focus is the study of gene expression in man-made cellular micro compartments, combining condensed matter physics, molecular biology, and materials science. We'll be hearing about this. He's now a tenured associate professor at the Weizmann since 2009. He's received many awards, the J.F. Kennedy and the Israel Parliament Prizes for Outstanding PhD Thesis, the Rothschild, the Fulbright, Burroughs Wellcome Postdoctoral Fellowships, and others. And this year, Roy has returned to the States and is currently the Elizabeth S. and Richard M. Cashin Fellow at the Radcliffe Institute for Advanced Study at Harvard University, where they bring together 50 fellows in all different disciplines to interact and to enrich not only the campus, but to enrich themselves. For his WALS lecture today, Roy will discuss the assembly of two-dimensional DNA compartments that are fabricated in silicon. I must say I've known Roy for quite some time, so I've heard about this many times, and we've seen really the culmination of many years' work in that recent science paper in August. These artificial cells that he's going to be talking about are capable of metabolism, programmable protein synthesis and communications, and offer many opportunities to better understand how cells work. So without further ado, Roy, thanks for joining us today.
Thank you very much, Steve, for the very kind introduction and for inviting me to give this talk. Thank you all for being here in this cold weather. And so today I will tell you about our ongoing work over the years, of the past roughly 10, 11 years in my lab back at the Weizmann Institute, trying to reconstitute gene expression in man-made compartments. The most important slide of this talk is presentation of the people. These are some of the students whose work I will be focusing on today. I'll start by describing the latest work, which is the work of PhD students Eyal Kartzbrun and Alexandra Tayar, who have developed the latest programmable DNA compartments, which allow us to look at gene expression in time in fabricated compartments. I will then go through work that we've done a few years ago, looking at the biophysical consequences of DNA attached to the surface at high density. What are the consequences of that? And this is the work of a number of people, Gabi Shemer, Yahel Atzmon, Eyal Kartzbrun, and Dan Bracha. Amnon Buxboim is my first PhD student who began working with me on this biochip approach, and he's now his own PI at the Hebrew University, bioengineering. And last but not least is Shirley Dowie, who is a staff scientist in the lab, who's a biochemist who leads our efforts towards reconstructing multi-component gene systems. And on the last work, my long-term friend and collaborator, Vincent Moireau, who's a professor at the University of Minnesota Physics Department, who has been creating amazing cell extracts which allow us to synthesize proteins under regulation. So the outline of the talk will be roughly 3 topics. I hope to cover them. One is the latest work, the 2D DNA compartments, which lead to emergent dynamics of gene expression in these on a surface, in fabricated elements. Then I'll describe to you some collective interactions in these DNA compartments. We'll zoom into the mesoscale of what happens at the DNA interaction level and DNA-protein level on a surface at high density. And the last, project into starting to look at coupling between expression and assembly of proteins into higher order complexes and see how we can reconstitute such kind of systems. By way of motivation, these are gene expression patterns emerge everywhere. They drive many, many processes in biology, of course. This is the language of biology. And for me, some of the guiding systems I've been thinking about all these years in trying to reconstitute gene expression dynamics in vitro sort of span between these two kind of very different systems. One, gene expression in space. This is a multicellular system. The morphogenesis of an embryo. This is already a scale of a millimeter, many, many cells here, as you all know. The striation, the appearance of gene expression patterns at the onset of symmetry breaking in space is a fascinating process. It involves encoding information in space, positional information. There are underlying circuits that guide the formation of these emergent patterns. There's collective decision-making, cells communicate, and there's transport of signals starting from a morphogen from one location throughout the remaining space here. And then following, there's growth and form, and so on and so on. So the notion here is there are many, many processes are coupled in biology. That's always the case. And in physics, we like to decouple processes. And it would be very nice to be able to realize a system which would allow us to investigate in a controlled environment the kind of processes which lead to gene expression patterns in space and isolate those parameters and study the fundamental principles underlying such a kind of systems. A totally different scale and circumstances is, for example, the assembly of a bacteriophage. This is my favorite T4 phage. It's the scale of 100 nanometers, and it's composed of a few dozen different proteins which make up its perfect body. It's an assembly line. It's precise. It forms inside a bacterium with high yield. And the main question, despite many decades of research in this system, is why is it so precise? Why is it synthesized with high yield? What is the thermodynamic landscape of a system that eventually assembles itself into such high precision structure? Despite the fact that we know the proteins, we know the order of assembly, we do not know, and it's difficult to study that in a living organism, in a living cell, why these kind of machines form with such precision. What are the underlying principles from the thermodynamic standpoint for these kind of complex formation? And again, the idea is that it would be very useful, we think, to be able to decouple the many processes that are going on associated with gene expression in a living cell or living organism, and to try to reconstruct gene expression patterns in time and space or in formation of complexes and so on. So this is general motivation for thinking about reconstituting gene expression in man-made compartments. In that sense, it's no different than reconstituting other cellular and molecular functions in vitro, from the study of enzymes, DNA, proteins, membranes. All of these functions have been reconstituting, but to reconstitute the dynamic nature and the regulation of how genes are turned on and off has been challenging so far. And so when we think about gene expression compartments, we also remember that they happen in highly compartmentalized areas in the cell. And this is, of course, the nucleus where mRNA synthesis takes place and protein synthesis just outside in the ER. And for me, this is a condensed matter problem. When you think about biosynthesis occurring in these kind of compartments, it is obvious that one has to take into consideration not just which genes are turned on and off, but also what is the physical state of the cell. How is it affecting the structure, the mesoscale structure? You see there's striation here. This is the membrane. All these black dots are the ribosomes. Nature has designed these kind of compartments in a purposeful manner to optimize the reactions differently in the different locations, and so on. And so even at the scale of a single bacterium, this is E. coli, work from Avi Minsky's lab at the Weizmann Institute, who studies how bacteria change their behavior in response to severe stress. The bacteria is able to condense its DNA and create a subcompartment within the bacteria volume. And again, it expels all the ribosomes, the DNA is inaccessible and enters a physical state of protection in which the entire metabolism drops down. And we need to remember that if we want to reconstitute a system which has anything remotely similar to that, we need to take into account that there's a huge amount of DNA, a few million base pairs per micron cubed volume, about 4,000 genes, and a very high protein concentration and proteins per gene on average. So these systems are very different with what we are used to think about dilute solution conditions for doing in vitro reactions. And we would like to be able to reconstitute not only the dynamic nature of gene expression, but also the physical state or the biophysical state of these compartments. It is very nice for me to acknowledge a landmark work that was done here at the NIH, which for me has been very, very important in my education when I entered my biology. This is the work of the group of Nierenberg and the landmark experiment he did with his postdoc then, Matai. And they were the first to basically, eventually in a series of papers published here, decipher the genetic code. And the way they did that is by making proteins in bacterial cell extracts. So it was a technical advance that allowed them to basically use artificially made RNA, mRNA, in this case the first one was a poly-U, and incorporate that in a cell-free reaction to synthesize a protein and see that you can have a labeled amino acid that eventually got incorporated into a protein, which they could eventually match the codon with the amino acid. That was the first codon that was deciphered here, and in a few years they were able to decipher the entire code. And so we've been using protein synthesis to basically try to create a man-made compartment where we can synthesize proteins. And since that period, cell extracts have been optimized and improved over the years for various purposes, from basic research to biotechnology applications. And today we're able to transcribe, translate in vitro with very high efficiency. Now, the earliest attempt to assemble a cell-free gene circuit, so to speak, was done when I was a postdoc with my advisor and colleague Vincent Noirot, and we worked together ever since. This was the earliest attempt, and it mostly taught us what cannot be done. What we did is basically try to reconstitute some sort of a lac operon, a synthetic system of the lac operon, in which the synthesis of one protein activate or stops the synthesis of another protein. So to reproduce the basic regulation of gene expression in a test tube reaction. So we made a few constructs which allowed us to basically, upon an allosteric transition with an inducer molecule, keep this kind of very small circuit either in the on state or in the off state, or induce it by will by adding a molecule to turn on the synthesis of a gene, just by creating simple cascades of reactions. And those, at the time, we used cell extracts that were available then, which were hybrid. So this would be T7 transcription and wheat germ translation. And since then, we've basically learned that you couldn't go very far by having hybrid system. Instead, you need entire endogenous transcription, translation. The entire machinery has to be preserved in order to generate use regulatory elements in a cell-free reaction. Now, how do we make the transition from one gene to a DNA program? So for many years, the paradigm for doing that is the artificial cell paradigm in a membrane, in a vesicle. The idea, eventually the sort of long-sought dream in this direction would be to encapsulate a genome or a genetic or a DNA program inside a vesicle, together with an initial pool of all the reaction mix, ribosomes, RNA polymerase, nucleotides, amino acids, energy resources, etc., to encapsulate the entire machinery inside the vesicle together with the genome and somehow bootstart the whole system, and eventually this will propagate itself. So if you encode the whole system, you can eventually lead to something that will replicate the DNA, synthesize new membrane, by some means divide the membrane, and so on. So generate a living system, a minimal cell, in a bottom-up fashion, so to speak. Of course, this is very challenging for a number of reasons. One, transport. It is despite the fact that a membrane can be incorporated with pores and materials can be transported, still a major challenge is transport, especially transport of bigger molecules. Another one is degradation. Protein degradation and mRNA degradation is essential for generating a source and a sink. In order to have dynamic gene expression, you need to degrade or dilute out what you synthesize after a certain time. Otherwise, you just clog the system and you cannot generate dynamic responses in a system. How do you degrade proteins in vitro remains a challenge. Encapsulation. If you want to encapsulate a whole genome or a synthetic DNA program in a vesicle, at that scale it becomes very challenging, not to speak of division. If you want to divide that kind of cell, that remains a major challenge. So we've chosen a different path, which is entirely artificial, is to basically use a surface approach. Rather than encapsulating the reaction inside the vesicle, we decided to immobilize the DNA templates on a surface, create a little nucleus, if you want to think about it this way. Instead of having a contiguous molecule which codes for all the genes, we just chop them into pieces and put them on a surface in a designated region. And then you can synthesize proteins locally, first transcription, then translation. Some of these molecules will diffuse and into the solution. Some of them can be brought back to the surface for further functionality. I'll show you that later on. And this is the whole concept. And we started out with this very vague notion and slowly built all our toolbox to enable that. And so the first step was to actually develop a biochip. Now, biochips, of course, were introduced in the early 90s, and they're used today as well as analytical tools for basically reading out the content of a nucleic acid solution, or for other reasons as well. Here we had to optimize a custom-made biochip platform, a material which would allow us to immobilize different kinds of functions on a surface, and in particular, localize DNA templates for protein synthesis. And that took a number of years to develop. This was the first paper, and then we had another step taking this whole realization into the electron microscopy technique. But the basic idea is that you have a photosensitive layer. It's a monolayer, which is comprised of molecules which are biocompatible and at the same time photocleavable. So you shine UV light through a mask, you deprotect the surface, you designate where you want to immobilize molecules, and you can do that without further details. You can basically create dense regions of DNA. And when I refer to DNA, I'm typically talking about 2 kbp DNA molecules that come from solution, bind where we designate them to do so, and they form highly dense structures, highly dense areas of DNA. Typically, the local concentration here matches that of E. coli. We have a few megabase pairs per micron cubed right there, which means that the typical distances between these DNA polymers matches the size of enzymes. 20 nanometers, typical sizes between these DNA polymers, and we can control their density. This is just an artistic representation of projecting an image onto the surface. This is a DNA carpet or a DNA brush whose density is varied in space, just a projection of a nice image. You see the scale, 50 micron, and in this, it's a fluorescent image, so we have little fluorophores attached to the DNA, so we know exactly where they are. So this is, in essence, the technique, and we spent a number of years investigating the properties of these DNA brushes, but instead I'll now focus on the encapsulation of these kind of DNA brushes into compartments, and this is the recent work, the most latest recent work we published. What you do now is carve, and you take a silicon device, coated with a thin glass coat, which allows us now to use our biochip approach. Our biochip approach is functional on glass. What you do now is immobilize DNA in these, at the center of these structures. So you have a circular reactor which is fed by a capillary. And this capillary is connected to a main flow channel through which the entire cell-free reaction is delivered. So you can see this is done by having a sort of robot which places individual drops on these pre-patterned regions for deposition of DNA, so the cells can have the same DNA makeup, or they can be different, they can have different kinds of DNA content, and so on. So this is the flexibility. So you spot the drops and then put the DNA, you wash away unbound DNA, you close the device and you flow in a reaction. And the reaction has everything in it. The ribosomes, the RNA polymerase, the nucleotides, amino acids, everything. It's cell extracts. And this scenario allows us for the first time to start to see emergent dynamics. Just by encoding different genes, we are now able to program oscillations, steady-state expression, self-regulation. This is in a nutshell what I'll show you in the next few slides. The simple construct of having DNA in this compartment introduces ability to maintain gene expression in steady state by a scenario of transport. And this is the scenario. So this is synthesis of GFP from its DNA right here. What you see, the DNA is entirely, it diffuses, it doesn't bind back to the surface. And what happens basically, because these are dead-end reactors, there's no connection right now, and these are extremely thin. The compartments here are really 2 micron thick, while this channel is extremely wide and thick. And so there's a mismatch in terms of the flow between this channel and this dead-end reactor in such a way that the only transport in and out is by diffusion. There is no flow inside out. Once it's filled, the only way to transport material and fluid in and out is by diffusion. So what happens is that the ribosome and company diffuse in, start to synthesize mRNA protein, which then diffuse throughout this entire reactor. And then once in a while, they find this little hole here and they exit here. And once they reach this output, they just get flown away. So that sets up a gradient of concentration. We have a source and a sink. And because this is only diffusion, the only solution to this is a linear gradient, a linear concentration gradient of product, in this case GFP. Now the interesting part is that by construction, because this is a 2-dimensional object, it's just like a pancake, and this is a linear object, a capillary, you can think of it, and this can be solved analytically, as basically this is a capacitor, electric capacitor, analogy to a capacitor. You charge the capacitor, you accumulate the concentration, you build a concentration, and then you discharge it through a resistor. This construction, the time scale, how long it takes to evacuate this volume, basically is determined by the product of the RC, the capacitor, and the resistor, giving you an effective lifetime. Think about it now as an effective lifetime of your proteins. Whatever you make, synthesize here, has a finite lifetime, which entirely depends on geometry and the diffusion constant of what you're making. So it's R squared, R is the radius of this compartment, D is the diffusion constant, W is the width of this capillary, and L is the length of the capillary. So it's a geometrically determined lifetime of whatever you synthesize. And this enters into everything we do. So this is showing you just GFP synthesis. The red squares are the DNA fluorescence. You see there's this synthesis of the same GFP has a very different kinetic signature. You see they all reach a steady state at different values. The values are entirely proportional to the length of the capillary. What happens basically is that it takes longer to kick off. You see it takes longer to get in. But once you get in, it takes longer to evacuate the whole reaction, and therefore the steady state value is higher. Very, very simple mechanism of transport, which now enters into the dynamics of gene expression, and you can program non-trivial reactions. And what I'll show here without going into detail is basically a number of very simple circuits, single gene, one which has an autocatalytic and one which is self-repressed genes. These are all based on E. coli and bacteriophage constructs, so the details I'm not going to go into. These are very simple programming of circuits and a feedback circuit with A and B, each one activating or repressing its neighbor. And just to give you an impression of how dynamics of a very simple ensemble of circuits unfolds on one single chip. We have about 100 compartments running in parallel, and you can vary on the same chip, and you can vary the DNA content of the reactions. Here, look at just no regulation synthesis of GFP, an autocatalytic reaction, a gene which activates its own synthesis. So you see this is a very different dynamic signature than this. This is an autocatalytic reaction. There's a very sharp threshold for activation before this kicks in. It has a very different signature. Likewise, a self-repression, a gene which shuts off its own synthesis. It does reach steady state, not at zero, because the system is able to modulate itself through this evacuation mechanism. And you see here, too, the dynamic signature compared to an unregulated GFP synthesis is exactly the same. The colors correspond to the length of the capillary, which changes the lifetime of the proteins. So this is long capillaries, this is short capillaries. If you go to 2 genes, it's very, very rich. You can go between emergent oscillations. You see, these are oscillations. We don't move anything by hand there. They are just encoded in the gene circuit. There's the complex timescales emerging in this kind of network to allow a regime for emergent oscillations. The oscillation cycle depends on the geometry. These are the same colors, the length of the capillary in this case. So the shorter the capillary, the faster the oscillation. We control that by geometry. And also the amplitude goes down. You can go by the same topology of circuit, just change parameters, like more repressor or more activator can take us between emergent oscillation to a decaying oscillation and even to a single pulse. So we have an entire dynamical system emerging just by very, very simple gene regulatory circuits on the chip. And this is the first time one can look at a compartment which has localized DNA at the scale of a real cell, giving us something which starts to look like gene regulation. And so you can see where we can take this. Another step is we have the spatial component. So here we can send signals in space. So now we've separated the compartments into A and B, connected by a capillary. And you can see that this generates a pulse in space, not just in time, but there's a hierarchy of activation and closing down of the gene. See? This is just synthesis of a reporter gene. The next thing you can do is basically create a chain of communicating cells. So not only have each cell be connected through a capillary to the main channel, but each cell is connected to its neighbor. Now that creates a mechanism of dilution, not just through the capillary, but also stepwise dilution to neighbors. And the result of that, this is a GFP profile, is a morphogen. This is an exponentially decaying profile at the scale of a few hundred microns, stepwise. This is one reactor, one reactor, one reactor, and we have linear drops. Just like we have linear drop concentrations between the reactor and the main channel, we have a linear drop of concentration between one reactor to its neighbor. And overall, if you look far enough, this linear chain will give you an exponential gradient, a morphogen, being emitted from one cell to all the neighbors. And we can control the length scale just by geometry. Here, these variables correspond to the distances of the capillaries, the geometry. And you can use that to trigger signals from one location to another along the chain, which is the first step towards a sort of morphogenetic program. This is now not just GFP, but a transcription factor, which activates a gene located in a compartment downstream in space. So if this is the morphogen, this is the emergent gradient coming out from this gene. And now you can imagine that if you now put all the cells, not just, you can create feedback by having appropriate circuits being embedded in these and study the appearance of morphogenetic patterns. So this opens a whole a research program of looking at synthetic morphogenesis in these compartments, just by connecting them. So you can do one-dimensional change or two-dimensional arrays, and so on. So I'll summarize. We have a new mechanism for transport and turnover that allows us to implement long-lived and steady-state synthesis, which allows us to program. Very simple programs of 1 and 2 genes already are complex enough, and we still don't have a full analysis of all the kinetic profiles I showed you. This is work in progress now, but clearly this will become very rich. It's a nonlinear system, and even 2 genes is interesting and very rich. We can control basic cellular reactions by geometry, just by this time scales. In the next few slides, I will describe to you, go deeper into what happens here, into the sort of physics of the compartment, and I want to give you a flavor why it's important to consider the physical state of the compartment, not just which genes are being activated or shut off, but what happens at the interface between DNA. Neighboring DNA are very close apart, so they interact. And they interact in a non-trivial way. I'll show you this in a minute. And also at the interface with machinery, which tries to penetrate into the DNA brush, actually doesn't like to do so entropically. Whoops. Okay. Okay, there we go. So without going into details, let's just show you a couple of results which I think are very, very interesting and illuminating and taught us something about the collective behavior of DNA polymers on the surface. And to do that, we basically used our density gradients to create one-dimensional arrangement of DNA density on the surface. We can see where the DNA is because we label them with fluorescence. And what we do now is measure what is the height of the DNA as it extends away from the surface. You can do that by using total internal reflection microscopy, standard technique. And then we measured the ionic strength in solution. Because DNA is a highly charged molecule, it responds not only to the density, but also to its ionic strength. Basically, its counter ions are released to the volume of the brush, and they alter the extension. And the way this is seen is the following. What you see here is basically DNA brush on the x-axis and the extension, the collective extension of the brush. You see, and the colors correspond to the ionic strength. The point I'm making is that this is very, very responsive. You can go between an entirely stretched DNA brush at no added salt, pure water, to an entirely collapsed DNA brush at high enough salt. And in between, you have a very responsive brush to density along the gradient and to ionic strength. So by taking the DNA brush to an unphysiological regime with no added salt at all, we can expose some of its basic properties. And to show you that there is basic physics here, you can take all this data and just plot it differently. Instead of plotting extension versus density in different salt concentrations, you can plot the extension as a function of the ratio of these two parameters, density and ionic strength, and observe data collapse. This whole data has a very simple representation when looked upon as a function of the ratio of these two parameters. There's a power law here which indicates what happens here. In a nutshell, what happens is that there are basically two competing forces here. The entropy of the DNA, which wants the DNA polymers to be compressed, because that's where the entropy is maximum. But there's also entropy of the counter ions of the DNA. And when there's no added salt, there is a strong mismatch. All the counter ions of the DNA in the brush remain collectively localized to the DNA brush to maintain its charge neutrality. But in doing so, they create an osmotic pressure, driving water in to balance. And so what happens, the entire brush swells, and this is this regime. The competition between these two forces, one driving the osmotic pressure, driving extension, and one the entropic elasticity of DNA, driving compression, creates this balance which leads to this collapse. And in a nutshell, this is the physics of a DNA brush, a competition between osmotic pressure of the counter ions, which are released, and the entropic elasticity of the DNA. And this is a manifestation of a collective effect. It has to do with the interaction of DNA segments in the brush. Interestingly, the physiologically relevant condition where DNA is already a neutral polymer because it's highly screened, you have enough salt there to screen all the electrostatic charges, is here in a very, very small piece of this phase diagram. The DNA, when it's neutral, the DNA, in fact, is a very, very special polymer. It's a stick. It's very, very stiff compared to other polymers that we can synthesize, man-made polymers. It's a very charged molecule. And as such, it turns out, this work revealed, that DNA, even though it's very, very, very dense, can accommodate many, many DNA segments in the same volume because it's a very, very rigid molecule. So my point here is that we've learned something fundamental about the DNA properties by being able to synthesize, to assemble these DNA brushes. The next step is to basically ask, what do macromolecules, either DNA molecules or RNA molecules or proteins, how do they penetrate into the brush? And the answer is, as long as they're large enough, they do not like to be in the brush. This is an experiment where we've labeled the DNA gradient. This is a gradient of DNA. Here, this is this axis, the density. And this is a turf measurement of RNA polymerase trying to penetrate into the brush. We do this by measuring its turf signal. It's labeled fluorescently, so you only see molecules close to the surface, and you see this black region is exclusion from the brush. And the denser the DNA, the more molecules are excluded. This is what this curve shows you. So in a nutshell, the formation of these very dense regions on the surface excludes DNA molecules, excludes macromolecules, other DNA molecules, and proteins above a certain cutoff. Now this has consequences for how machinery, transcription machinery, respond to what you put on the surface. They respond to the density, and they respond to the orientation and position of the genes, just as they would, presumably, in E. coli or in other cells where they sense the local environment. This is a very primitive demonstration of that, but it's a very clear one. And what we've done here is basically take genes, or reporter genes, and just increase their density. And we have a curve, a very simple curve of output versus gene number saturating at a certain value. There's no depletion of reaction components, nucleotides, RNA polymerase is in huge excess. And yet, if you just flip the orientation of the same gene upside down, you get much more protein out. The same gene, this is just luciferase reporter, showing us clearly that what determines the overall synthesis from the brush has to do with its properties. In this case, the density and the orientation dictate what happens inside the brush. It's a very clear demonstration. There's more data than I can show right now. And the main hypothesis and questions that arise from this kind of work is that, can we somehow do better by introducing crowding and changing the condition inside the compartment? Can we actually do much better than we're doing now? Because right now, proteins are being rejected from the brush. And of course, the cell is a much more crowded environment than our cell-free system. It's significantly more crowded than we are, and there are probably reasons for that, which have to do with why we get much less proteins that we could, as we see by this exclusion. Okay, so in summary of this part, and there I'm coming to my last part, is that DNA brush exhibits collective properties. Macromolecules are rejected from the DNA brush, and directional transcription leads to some sort of confinement. The fact that enzymes are able to synthesize more proteins when they're pointing into the surface suggests that there is a confinement effect of enzymes within layers in the brush, such that the local concentration of enzymes reading the DNA is higher than if you just flip the orientation outward and enzymes are ejected out. So in a nutshell, we need to get closer to the working point of E. coli. Not just the gene contact, but the physical state of the compartment. Oops, another one. Okay. Okay, very few slides to sort of give you another direction we are pursuing is to try to look at the coupling between protein synthesis and assembly on the chip. And we took our favorite phage, T4, and began to work to isolate some of the well-known proteins along the pathway making the phage tail. So this is a pathway from one of the books. It's well known. What we've done is taken and synthesized a couple of genes. One gene is this crystalline tube-making protein, and another one makes this capping ring. You synthesize them in vitro, and they indeed make beautiful crystalline tubes and beautiful nanorings. You can generate them with high yield. But this is all done in solution. It doesn't reconstitute the native process, the natural process occurring inside the bacterium. And if you just take two of the two genes and simultaneously synthesize the proteins, you get something which may or may not occur inside the bacterium, but we didn't expect that. We get an ensemble of crystalline tubes and donuts which seem to have appeared out of the co-synthesis and assembly of these two proteins. This was very rare and uncontrolled, but this was done in solution. So to make progress in this sort of direction, we used our surfaces to guide the assemblies back to the surface. And this is a demonstration of putting a gene, coding in this case for fluorescent proteins, and next to it, antibody traps. So these are affinity tags fused to the proteins for which we have antibodies immobilized here. And what this experiment shows you that we can generate by combining synthesis on the surface and in solution, we can generate patterns of proteins synthesized on the surface and use those to basically template assemblies. To go further, we use the biochip and ask ourselves, can we reconstitute an assembly reaction, synthesis and assembly, without any intervention, such that the readout will be directly on the chip, as if this was happening inside a bacterium. No intervention. So we use an extremely thin glass, about 10 nanometer thin glass, coated it with our biochip, immobilized antibody traps, and synthesized in situ this crystalline making protein from the phage. At low resolution, you don't see much. Low resolution, you don't see much. You go to high resolution, you see the formation of these nanotubes, crystalline nanotubes. You can see them here, one individual one, forming directly on the surface. The reason why we say form directly on the surface is by a control experiment in which we took pre-assembled tubes and put them on the surface, on the antibody surface. Not much was retained, suggesting that we have a nucleation and growth process happening right there on the surface. So I will end up by giving 3 slides of outlook. The next step is to basically scale up to about 100 genes and maybe a few 100,000 or 10,000 connected cells on a surface. Scaling up to 100 genes is going to open us the possibility to look at gene regulation on a larger scale, to ask questions about metabolism, homeostasis, and assembly of molecular machines. I've told you about our effort towards the phage, but one can think about synthesizing ribosomes and other molecular machines. Just reconstitute one after the other all the components, all the important components in a cell. Regulation, metabolism, assembly. Another, to go into a multicellular system would be, I alluded to, looking at a morphogenetic scenario in which we now have connected cells which communicate. They are triggered by morphogen. We can do that. And if you embed genetic circuits in these cells, you can start to see which kind of patterns leads to which which kind of circuits lead to patterns, and look at collective decision making and transport and so on. And last but not least, this opens up possibilities for these kind of artificial cells in technology and biomedical research, from synthesizing materials to sensing and drug screening, personalized medicine. You can think about designing systems that will ultimately not only sense whether a molecule is there or not, but whether it's functional. Because just like cells respond to the functionality, we can program our circuits to respond to function, not just existence. And eventually, this might lead to advanced engineering of metabolic pathways and so on. Last, of course, not by least, is the lab. This is the current lab. Our staff scientist, Dr. Shirley Daube, and the PhD students, some of his work, their work I presented. Eyal Karzbrun, who just finished his PhD, together with Alexandra Toyar, they're responsible for the last work I showed. Itamar Shani, I did not talk about his work as in physics. Ohad Vonchak works on multi-component systems. I did not mention his work. Dr. Dan Bracha just finished his PhD, worked on the properties of the DNA. These are master's students. These are former students and collaborators who have provided immense help over the years, and my long-term collaborator, Vincent Moreau. Thank you so much.
Since this is being recorded, we appreciate you going to the microphone when you ask a question. Identify yourself, please.
I'm Vittorio Sartorelli with NAMS. This is a beautiful work, really congratulations.
Thank you.
You have been able to recapitulate all the positive and negative feedback mechanisms that have been illustrated by your colleague Uri Alon and other people. I have a question though regarding your next enterprise in Drosophila. In bacteria, there is no epigenetics. There is no histones. Do you have any evidence that in a eukaryotic system, all the lessons that you have learned can be somehow recapitulated? And how are you going to go after the question of gradients that regulate transcription in a epigenetic system like Drosophila? Thank you.
Yeah. So to address gene regulation in a eukaryotic system would require, in my view, developing mammalian system, eukaryotic-based cell-free systems, which are entirely endogenous. There are beginning to emerge some systems of mammalian expression, but as far as I know, they're not endogenous. Namely, the transcription and translation are not endogenous. So to actually look at gene regulation at the eukaryotic level would require systems that need to be developed further. So like splicing and so on, that require different kinds of systems that we use today. I think there's enough room right now. We've just scratched the surface, being able to show possibilities. And for us, we are going to stay for some time with bacterial system because we have the language of promoters and regulatory system, which is rich enough in vivo and is certainly opening possibilities for us to study dynamics of gene expression, even though they're, of course, much simpler than any eukaryotic system. So for now, we're going to stay with prokaryotic systems.
So if there are no other questions, I'm going to thank Roy for all of us and very illuminating talk. There were many people who were watching, I'm sure, and remind everyone that there is a reception just beyond the doors here after the WALS lecture sponsored by FAES. Thank you very much for being here.